It is now my pleasure to turn today's program over to Belinda Lee from BD Biosciences. The floor is yours.
Thank you. It is my pleasure today to introduce Dr. Rafik Terra, the Scientific Director of the Special Hematology Department at Maisonneuve Rosemont Hospital in Montreal, Quebec. He holds a doctorate in biomedical sciences and immunology and a postdoctoral degree in the field of transplantation. He has completed additional training in multicolor flow cytometry to standardize diagnoses of hematological malignancies, including multiple myeloma, with EuroFlow. Since 2009, he has served as an executive in the quality of hematology laboratories for Maisonneuve Rosemont Hospital and as a scientific director of the clinical immunology laboratory. He is in charge of post-treatment assessment of minimal residual disease for multiple myeloma. Welcome and thank you, Dr. Terra.
So thank you for the introduction, and thanks also to give me this opportunity to give this talk about study concerns the evaluation of the impact of standardized 8-color flow cytometry protocols made by EuroFlow on the diagnostic accuracy of acute leukemias in general and poorly differentiated acute leukemia in particular. So I would like to refresh our memory on some important notions concerning the diagnosis laboratory for the hematological neoplasia, motioned by the last version, revised edition of WHO classification for 2017. So first of all, there is no single gold standard by which all diseases are defined. Then the availability of this information concerning immunophenotype and genetic means that it's now easier to establish consensus definitions than it was when only subjective morphological criteria were available. At the end, many diseases have an immunophenotype so characteristic that is essential or nearly essential for the diagnosis. So you can notice the importance of the multi-parameter analysis on the diagnosis and also the importance of immunophenotyping on this file. So are there anything new on the recommendation concerning the flow cytometry in this last edition of WHO? I can tell almost nothing except for the classification of acute leukemia. For example, you can see that the entity blastic plasmacytoid dendritic cells neoplasm, which was previously under the entity of acute myeloid leukemia, is now totally separated and it's alone. And then you can see that the introduction of the entity of early T-cell precursor lymphoblastic leukemia, which called ETP-ALL, which was absent in the previous version and actually introduced under the T-lymphoblastic leukemia lymphoma. So why these two entities and what is the common point between them? The thing is that these two entities are rare diseases and also they have a very bad prognosis. And also the diagnosis of these two entities relies on strict immunophenotyping criteria by flow cytometry and immunochemistry for BPDCN, blastic plasmacytoid dendritic cells neoplasm, and also based on flow cytometry exclusively concerning early T-cell precursor lymphoblastic leukemia. Because until now, we cannot find a genetic signature concerning these two entities, so the only way to make the diagnosis is by flow cytometry. So acute leukemia is a heterogeneous group of diseases reflecting stem cell of origin with their characteristic of potency and plasticity, and thereby leukemic cells may show early or minimal differentiation. So concerning some entities, including poorly differentiated acute leukemia, for example, for AML-M0, or immature PBDC neoplasm, or ETP-ALL, or pre-timing T-ALL, we can have some stem cell, myeloid, or lymphoid antigens expressing on their leukemic cells. So thereby, we can make confusion between these entities, for example, between early immature lymphoid or PBDC acute leukemia and acute myeloid leukemia with minimal differentiation, which is called commonly AML-M0, if proper immunophenotyping panels are not routinely performed. So to evaluate if the use of standardized approach has the potential to improve the diagnostic accuracy of rare entities like PBDCN and ETP-ALL, we use the standardized EuroFlow approach to reanalyze cases of poorly differentiated acute leukemias. In other words, so what is the impact of non-standardized laboratory-developed panels on the accuracy of acute leukemia diagnosis? So to be able to reanalyze all cases, we took advantage from the BCLQ, which is the Quebec Leukemia Cell Bank, which provides us of fiscalized high-quality specimens. Also, with the computerized database, includes all laboratory tests as well as the corresponding clinical data associated with each specimen. So with these stored cells, we will be able to reanalyze with standardized panel the flow cytometry and compare it with the previous diagnosis based on multi-parameter analysis which have done before. So our objectives were, the first primary objective is to establish the extensive immunophenotypic profile of cases of acute myeloid leukemia with minimal differentiation, acute myeloid leukemia with myelodysplasia-related changes, or acute leukemia of ambiguous lineage. The second objective is to identify cases corresponding to PBDCN and ETP-ALL and describe the associated cytomorphology and immunologic expression patterns. Then to compare the survival data of different groups of patients according to their immunophenotype and the treatment protocol used. Finally, to compare the mutation profile of the different groups of patients by next generation sequencing using an AML-related gene panel. So we selected 49 cases collected in the last 15 years, including, as previously mentioned, AML-NOS with minimal differentiation, or AML-M0 by A5B classification, AML with MDS-related changes, which can include any one of the NOS acute leukemia, and acute leukemia of ambiguous lineage. We excluded all the leukemias with cells having monocytic and or myeloid differentiation demonstrated by cytochemistry or by flow cytometry, and we kept 6 cases of them as controls. So here you have a table of total number and characteristics of reanalyzed patients. So you have 49 patients with samples concerning patients with a median age of 62 years old. 28% are male, and the initial diagnosis is reported here exactly as reported on the BCLQ documentation. So we have 19 cases as AML with myelodysplasia-related changes, 9 cases as AML-M0, 5 cases as AML with minimal differentiation, 1 cases as AML without maturation, 4 cases as acute and differentiated leukemia, and some cases as mixed phenotype acute leukemia, 2 B and myeloid, 3 as T and myeloid, 1 case B and T, and 1 case NK myeloid, which called also not otherwise specified. 2 cases RT-ALL and 2 cases RB-ALL. 32% got induction therapy, 15% without therapy, only with palliative or supportive care, and 2 cases with a known therapy. So the cells were reanalyzed using the 8-color immunophenotyping protocols after thawing, and using at the beginning the acute leukemia orientation tube, and then AML-MDS panels if it concerned the AML acute leukemia. If there is any evidence of the presence of plasmatoid dendritic cells, we added 3 specific markers, which are TCL1, BDCA2 and BDCA4 as specific antibodies for plasmacytoid dendritic cells to confirm this entity and to confirm the nature of PBDCN leukemia. And finally, cases shown in a population with a B or T phenotype were further characterized using B or T ALL panels. Events were required on a BD-FACSCAN2 instrument, and data was analyzed using InfiniSight software. Cytomorphological examination of the identified samples were reviewed. DNA sequencing will be performed on all specimens. Only one is already done with next generation using an AML-related chain panel. Clinical data including demographics, blood count and diagnosis, extramedullary manifestations, treatment received, and survival were retrieved from the BCLQ database if it's available. So here you have the acute leukemia orientation tube, which was used on all the specimens. It's the first tube we have done to confirm the nature of the leukemic cells. So if we have this tube is composed for 8 antibodies specific for each lineage. So if we have the presence or positivity of cytoplasmic MPO, we will perform the AML-MDS panel, which composed by 7 tubes. And if there is any evidence of the presence of plasmacytoid dendritic cells, so we add the 3 specific markers. Then, if we have the positivity of the 2 specific markers of B cells, cytoplasmic CD79A and CD7019, so we performed the BCP ALL panel. And finally, if we have positivity of the 3 specific markers of T cell lineage, including cytoplasmic and surface CD3 and the expression of CD7, we performed the T-ALL panel. Just take a look on the most used panel on our study, which is the AML-MDS panel, composed by 7 tubes, and each tube is specific for lineage. For example, the first tube is concerns the neutrophilic maturation. The second tube concerns the monocytic maturation. The third tube, erythroid maturation. The fourth and fifth tube concerning aberrant expression of lymphoid markers or other aberrancy or markers correlated with mutation or translocation. The sixth tube concerns the megakaryoblast, basophilic, and plasmacytoid dendritic cells. And the last tube is to confirm the megakaryoblast leukemia and systemic mastocytosis. So our results show that among the 49 cases we analyzed, we modified 18 cases, so 37%, and we refined 5 cases, so 10% of our selected samples. The refined cases, we defined the refined cases as modification without impact on treatment. It means that we didn't change the nature of the leukemia, we just refined the classification. So cases in the beginning, which were B-ALL, become pre-B-ALL for 2 of them, common B-ALL for 1 of them, and precursor B-ALL also for 1 of them. And the other one as cortical T-ALL. So you can see that we still have the B-ALL or T-ALL leukemia, but we refine the classification, which can be helpful for the follow-up, for example, for the evaluation of minimal residual disease. For the modified cases, we have like 18 cases would have had a different diagnosis. So 37% of these cases, 1 case or 1 sample showed a clear presence of megakaryoblast population. 3 samples revealed previously unrecognized cases of early T-cell precursor, and also 10 cases showed the presence of plasmacytoid dendritic cells, including 2 typical cases of mature PBDCN, expressing CD56 marker, and 8 cases containing different proportions of plasmacytoid dendritic cells, which are CD56 negative with or without another component of cells like monocytic cells or even abnormal B cells. And finally, we unexpectedly demonstrated that with EuroFlow panel, we can confirm the presence of high proportion of monocytic components previously not recognized, even with morphology and cytochemistry and with laboratory developed panels. So if we go through these different cases about clinical history and immunophenotype and also treatment, concerning the 2 cases of typical PBDCN with a high proportion of plasmacytoid dendritic cells, 81% and 88%, the initial diagnosis was, for the first one, TALL and AML. So we changed it to typical PBDCN. These cases was treated as AML and ALL induction without transplantation, and the survival was 310 days. The second case was initially mixed acute leukemia, NK myeloid, with a known initial therapy, and the survival was of 181 days. Then we have all the cases with the different proportions of plastic plasmacytoid dendritic cells, with or without monocytic cells, and even with abnormal B cells. So the proportion of PBDC was from 6% to 22%, with a high proportion of blasts with minimal differentiation. So the initial diagnosis were AML with myelodysplasia-related changes, or acute and differentiated leukemia, or AML with minimal differentiated M0, or also AML NOS not classified. So among these cases, only 3 got treatment with AML induction, one of them ALL induction, and the only one still alive is the one treated AML with transplantation. All the others, as you see, passed away very fast. Then you have cases with a significant proportion of monocytic cells. So initially diagnostic as AML NOS, AML with minimal differentiation M0, or AML with myelodysplasia-related changes. 2 of them were treated with AML induction and transplanted, and only one still alive. And finally, you have a case as acute megakaryoblast leukemia, because we showed a clear population of megakaryoblast leukemia, and this case got only a palliative support no treatment with 10 survival days. And finally, the 3 ETP ALL with very bad, described as with a very bad prognosis. For the first one, it was AML NOS M0 with AML induction and transplantation with only 300 days of survival. The second one was TALL, but not as early TALL, got ALL induction only with 43 days of survival. And the last one was described with mixed phenotype acute leukemia, T myeloid, got AML induction with transplantation, and it's the only one still alive. So before showing you the immunophenotypic characteristic of each entity and how we detected this population, I just would like to show you the perfect preservation of the immunophenotype for the cryopreserved cells after cycle and after thawing compared to fresh cells. So a part of the granulocytes which disappear because of the cycle, you can see that we maintain or we preserve a perfect expression of different markers, like you can see here the 34 expression, here the 117 for its intensity, and here the monocyte for the expression, heterogeneous expression of CD117 and expression of HLA-DR. Also, you can see different markers as CD13 with its bright intensity, and also the profile maturation of granulocytes here on the fresh cell, and on the ficle cells, on the residual granulocyte, we can keep a perfect profile maturation for granulocytes, also for monocytes. The intensity of CD64 is perfect. The heterogeneous expression of CD14 is perfect. And also for other markers like CD7, CD38, CD15 on the red blast cells, on monocytes and on granulocytes, and also for CD123, which is important for detection of plasmacytoid dendritic cells. So you can see clearly that we really preserve a perfect immunophenotype with its expression and intensity for all the markers, even if the cells are cryopreserved, cycled, and thawed after that. So the first case is concerning megakaryoblast leukemia. On the fresh cell, the morphology, you can see here, there is no evidence of megakaryoblast cells on this slide. The MPO was negative, and here you have the cells after post-thawing after cytospin. And with the immunophenotype, you have on red here the blastic cells expressed and partially endeared the CD117, expressing CD34, so it's immature cells, and clearly express a high level of CD36, 42A and 61, 41 and 42B also. So these markers are specific markers of megakaryoblast cells. So to confirm or to trying to find another evidence of megakaryoblast nature of this leukemia, we performed the next generation sequencing, and interestingly we found potential pathogenic mutation including TET2, KMA2C, RANKS1, and GATA1. GATA1 has interestingly been reported in acute megakaryoblast leukemia. So this mutation gives a powerful and supports the finding of flow cytometry. Then I just show you here how with the EuroFlow panels we can detect the monocytic component, because we find a proportion of specimens with previously unrecognized monocytic component, and we demonstrated here that these cases contain a significant proportion of these monocytic cells. So here you have the APS, which is the automatic population separator, which is the combination of the 33 antibodies containing in the 7 tubes, and with the different clusters you can notice the presence of granulocytes in green, monocytes in blue, blasts in red, and lymphocytes in orange here. This is normal bone marrow. So with the expression of CD117, HLA-D and HLA-DR, we can see the pathway of the maturation of monocytes from blast to monocytes by losing the CD117 and preserving the expression of HLA-DR, compared to the granulocytes which lose the CD117 and also the HLA-DR1, the HLA-DR. So here you have a case of acute myelomonocytic leukemia, AML-M4. So inside the tube of monocytic maturation for the AML-MDS panel, you can see that with 117 HLA-DR, we can, at the beginning, demonstrate the presence of this monocytic cell. Then with specific markers like CD36, with its high expression or bright expression of CD64 here, heterogeneous expression of CD300 and also of CD14. And even plus, we can see that we have a pathway of maturation with the CD300 and 14. With the dotted line here, it's a normal pathway maturation of monocytes with expression at the beginning of CD14, after that expression at the end of CD300. In this case, you can see that this pathway is inverted. We have at first expression of CD300, and then we have expression of CD14, reflecting in mutation, for example, like NPM1. Here, another case of acute monocytic leukemia with the presence of monocytes in blue, expressing HLA-DR 36, bright expression of CD64, heterogeneous expression of CD300, and also for CD14. So that is the way how we detect the presence of monocytic components in each of our specimens. So this is one of the cases we modified with a high proportion of monocytic cells. So at the beginning, on the fresh cells, with morphology and cytomorphology with butyrate and double esterase, we can see that we demonstrated that we have less than 10% of monocytic cells. Using EuroFlow panel on those cells, we demonstrated that we have 44% of monocytic cells, which are HLA-DR, high expression of CD64, and expression of 14, and heterogeneous expression of CD300. Second case, with the initial diagnosis, we demonstrated by morphology and cytochemistry, and even with laboratory-developed panels, that we have only 2% of monocytes. By EuroFlow panels, we clearly demonstrated that we have more than 25% of monocytes inside this specimen with CD64 and 14, and also with the expression of CD300. Concerning the blastic plasmacytoid dendritic cells neoplasm, we have the two typical cases we identified. So we modified clearly the diagnosis from AML N0 and AML NOS. And we can see here that our red cells, blast cells here, express HLA-DR, express CD7 with high level, express CD56 and TDT, a bright 123 with CD4, and we confirmed the nature of these cells with specific markers like cytoplasmic TCL1 and with CD300 here. Then concerning the immature cases containing plasmacytoid dendritic cell CD56 negative, which can be mixed with other populations, we can see here that these cells express HLA-DR, express CD34, express a bright CD123, 36, and also CD304, CD303, and partially expressed CDTCL1, cytoplasmic TCL1. So we have here clearly demonstration of the presence of plasmacytoid dendritic cells using all these markers. Interestingly, we tried to find a way to identify the cells without using the specific markers of plasmacytoid dendritic cells because they are not present on the AML panel of EuroFlow. So we find that the level expression of CD123 is useful to detect either plasmacytoid dendritic cells and also to discriminate between typical CD56 positive and immature CD56 negative plasmacytoid dendritic cells. You can see here that on immature PBDCN, the CD123 is stronger than the one expressed on mature cases of plasmacytoid dendritic cells, which are CD56 negative. Positive, sorry. So finally, to conclude, these results support the major role of comprehensive immunophenotyping panel in the diagnostic accuracy of acute leukemia by multicolor flow cytometry. So using limited immunophenotyping panels, even with the global characterization, as we showed on the specimen, cryopreserved and the cell bank, including cell morphology and cytochemistry, we demonstrated that incorrect diagnosis may result. So extensive immunophenotyping using the EuroFlow approach refines the diagnosis with cases of with very bad prognosis like ETP-ALL, CVDCN with mature and immature cases, and unexpectedly we demonstrated the presence of a significant monocytic component, even without using of morphology and cytochemistry. So the use of standardized approach to flow cytometry can lead to more precise diagnosis of poor prognosis leukemia subtypes like ETP-ALL and BPDCN as expected, and has the potential to change the therapeutic approach with potential improvement of clinical outcome for some of these patients. So finally, we have shown with a megakaryoblast case that NGS can be used in the clinical laboratory to refine the diagnosis of acute leukemia.
At this time, we would like to take any questions you might have for us today. To ask a question via the web, click the Q&A button in the lower left-hand corner of your screen. Type your question in the open area and click Submit.
Thank you, Dr. Terra, for a great presentation today. Before we ask any questions, we're going to be pushing out a brief poll. It'll take about 20 seconds, so if you wouldn't mind answering, that would be great. Dr. Terra, we have one question that came through. The first is, how do you determine the depth of characterization to do? Is there a recommended approach to stratify the use of EuroFlow tubes to both minimize reagent cost, but also to maximize accuracy?
Okay, thank you. How to determine the depth of characterization to do? Usually, we always use the recommendation on this file of hematology. So we based on WHO recommendation, and then we continue with what we are using in our laboratory. As we are talking about EuroFlow approach, so we have to follow the EuroFlow approach because they built these panels. So they built screening tubes first, so we don't have a choice to use the screening tubes to know in which way we have to go. So if we are talking about acute leukemia, so we have to do the allot tube to know which cell line we have inside our specimen, and then based on the results, we will use the characterization tube. So if it's acute leukemia, so we will use the characterization tube of acute leukemia, and if it's lymphoma, we will use the characterization tube of B-cell lymphoma or T-cell lymphoma or NK cell lymphoma. For acute leukemia, so we have the AML acute leukemia characterization tubes, or ALL, T-ALL or B-ALL. So, we have just to follow the recommendation based on the results we have, and EuroFlow approach make this easy because of the screening tubes. So usually, for example, for our lab, previously we don't have this approach of like screening, or so we have we had like directly one panel in which inside we are using several antibodies based on literature, and then we repeat and we repeat and we repeat until we have all the answer we want to have. And with the EuroFlow approach, the advantage is that you have one tube as a screening tubes, help you to know which way you have to follow to do your characterization. For the approach, is there a recommended approach to stratify the use of EuroFlow tubes? EuroFlow approach is in itself a gain of time and money. So if you use the EuroFlow tubes, we are, gaining time, and you are gaining money. And as we are, again, I repeat, a lab, we used previously homemade panels or laboratory developed panels, and we switched then on this approach. So we realized that we really gain time because we don't have to repeat several times by adding some antibodies we don't have in our first tubes we used. And then gain of money. It's true that there are too much antibodies in these panels, but all these antibodies are titrated and specific for something. So at the final, we realized that we are really minimizing the reagent cost by this approach.
Thanks, Dr. Terra. So a second question that's come through is from Norma. She asks, could you tell me more about the marker APS1 used with the CD33 to better discriminate the populations?
Sorry, can you repeat? CD?
CD33.
33. In which panel?
She asks, could you tell me more about the marker APS1 used with CD33 to better discriminate the populations?
APS is not a marker. It's like automatic separation population. So.
Okay, perhaps we can answer that one offline and get more information from Norma. The second question that we have is, how well does EuroFlow perform in cases of lymphoma? Are these disease categories not well identified by this approach? Are there disease categories not well identified by this approach, for example, Hodgkin's?
Okay. For lymphoma, based on WHO recommendation, you have some entities which, are characterized, and we don't have choice to be characterized usually by flow cytometry, like CLL or mantle cell lymphoma. So for this entities, EuroFlow is really useful to help you to very well discriminating between, for example, CLL and mantle cell lymphoma based on several markers, and also based, they confirm that some antibodies are more useful than others based on the APS. They are used for automatic population separator. They demonstrated that, for example, CD200 is and the CD79B is more useful than CD23 to discriminate between CLL and mantle cell lymphoma. So that is an example. Other thing, it's very useful to diagnose this hairy cell leukemia, and also discriminating between hairy cell leukemia and marginal zone lymphoma, between also follicular cell lymphoma and CLL. So you can see that we have many choices of way to precisely diagnose all these entities. The only thing we still not able to discriminate, for example, between marginal zone lymphoma and, for example, lymphoplasmacytic lymphoma. It's still like difficult to discriminate between these two lymphoma. And also in pathology, it's not there's no like 100% consensus between pathologists to discriminating between these two entities. So I can tell you that it's, of course, it's very useful. It provides more information, for example, for, as I have said, to discriminate between CLL, mantle cell lymphoma, between hairy cell leukemia, marginal zone lymphoma. But for example, for Hodgkin lymphoma, no, there's nothing, more benefit with these panels to diagnosis for Hodgkin lymphoma.
Thank you. Another question we have is, have you had occasion to use the SST tube? Say, for example, with CSF. Has it been useful?
Yes, of course, the SST tube is extremely useful. Also because according to our experience, previously, we have made one tube to for example, characterizing specimens as CSF, which are specimens with very few cells. So once we switch to SST tube, in which we have like markers helping us to, characterizing which populations are inside the cells, the most important. So we really realize that there's much benefit. And also, the benefit is increased if you use, for example, Infinicide software, which helps really more and more because it helps you to remove all debris easily to be able to target the live cells inside. And also the techniques is very important. The tube in which we collected, for example, the CSF. So if you use the SST tube without, for example, conserving or collecting your specimen inside, for example, the transfix tube in which you have a preservative, conserving the CSF during several days at 4 degrees. So you will really lose your cells very fast. After 30 minutes, you will start to lose your cells. So you have several points. Yes, SST tube is very useful, but you have one thing that's very important is the collection tube in which you have to preserve your specimens, which is transfix. And also, according to our experience, Infinicide software is really helpful also to do the analysis.
Excellent, thank you. Another question. In your hands, how well have the screening tubes correlated with the final clinical diagnosis? Have you seen any cases where a myeloid or lymphoid read from an ALOT tube turned out to be something else?
Thank you. But as I said, the screening tube, if we talk about, for example, acute leukemia, the ALOT tube can help you to diagnose like 80% of acute leukemias. So with the ALOT tube, you will be able to tell if your leukemia is myeloid or lymphoid or like undifferentiated or minimally differentiated. So after that, once you have this question, you have to do the characterization because if it's B cell nature, you want to know at which stage are your B cells are blocked, or if it's T cells, also the same thing. And if it's AML, because your cytoplasmic myeloperoxidase is positive, so you want to know which AML you have. And you have, 7 types of AML. So the screening tube usually correlate with the final clinical diagnosis. And sometimes you can start with one screening tube because according to your clinical information on your request, because usually we based on that to start something. So if your information, and we can see that several times is lymphoma. So you will start, for example, with LST for lymphoid screening tube. And finally, we realize, for example, that we have B cells or cells with the CD19 expression, but these cells don't express light chain at the surface of them, and they are like little bit CD45 dimer, which could happen also for lymphoma. But as these cells don't express kappa lambda, so we will switch toward another screening tube, which is allotube, and finally realize that we have acute leukemia and not lymphoma. So that's the way how to work, and the screening tube is supposed to be very helpful to guide you in which way you have to go, and also to confirm the information you have on your request.
Thank you. You mentioned the Infinisight software earlier, and I have a question around the Infinisight software. So how would analysis be performed outside of the Infinisight software, for example, through Factiva or other softwares? And then a follow-on question to that is, what might be the advantages and disadvantages of various software packages? If you could speak to that, that would be great.
Thank you. I mean, based on my experience, I mean, during all my experiences, my life in flow cytometry, I used really all the platforms of softwares to do the analysis, the free ones, the ones we have to buy. So you have a package of softwares. And in clinical file, I started to use the software provided by the companies, which like Factiva or other softwares, if people are using other machine than BD Fax. So I can tell you that the Infinisight software is really the strongest one. When you start to use it in the beginning, it's a little bit like disturbing, because our brain have the ability to use certain way with the gating, with the percentage, to do the analysis. But once you start to be very comfortable with it, you will realize that there is a huge benefit to use this software. It's really I mean, built, and people think about this software especially for clinical, and it's really adapted to and connected directly with the EuroFlow panels. So if you use the EuroFlow panels, I really recommend it strongly to use this software compared to Factiva. If you ask me to use Factiva to analyze this, I mean, my cases, I will have difficulties to do that, honestly. So I cannot see any disadvantage of Infinisight software unless for the price, but for other things, I think it's very strong software, and I recommended it strongly for people doing clinical analysis.
Thanks, Dr. Terra. Good information. So the last question that we have for today comes from Dr. Daniel Maza in Brazil. He asks, I have used TCL1 to the diagnosis of T prolymphocytic leukemia. You have mentioned it to be used in AML-MDS panel. What is the importance of TCL1 in AML-MDS panel?
Okay. TCL1, if I well understand, you talk about prolymphocytic T-cell lymphoma. So TCL1 is inside the TCL-PD panel. which is very specific for prolymphocytic lymphoma. And the few cases I have seen during my experiences for this T-cell lymphoma, they were positive for cytoplasmic TCL1. For the acute leukemia, TCL1 is not inside the AML panel, as you notice, because there's no really first utility and very important to be inside. You have bunch of other markers more important than that. The only thing is, for example, for our project, as we are investigating, looking for this entity of plastic plasmacytoid dendritic cells, and we want to confirm if we are missing or doing confusion between another entity and this entity of plastic plasmacytoid dendritic cells, we added the TCL1 based on the literature, because it's one of the antibodies helping to characterizing the entity of plastic plasmacytoid dendritic cells. It's not always positive, but it could be positive in some cases. So if it's positive, it will confirm to you, so you have to be careful with other markers, of course, that is really, or give you more confidence to conclude that it's plastic plasmacytoid dendritic cells. But it's negative, you cannot exclude that it's plastic plasmacytoid dendritic cells. So it's only but if you look at these markers, it's not present in the AML. So in our routine, we have it, because we have it in TCLPD also panel. So for some cases, when I have cytoplasmic MPO negative, and they have other evidence like HRDR positive, CD56 positive, or CD123 bright, so I ask people in the lab, technologists, to add the cytoplasmic TCL1 just to confirm, and also by curiosity to know if this leukemia is cytoplasmic TCL1 positive or no.
Thank you. Again, some great information. So we are out of time, and thank you so much, Dr. Terra, for a really great presentation, very informative, and thank you again to all the attendees. Just to let you know, we'll be sending out the presentation. You'll get it by email. And also look out for next month, we will be having another webinar. So we'll keep you posted with all that information. And again, thank you so much for joining, and have a great day.
Thank you very much.
Thank you to all our participants for joining us today. We hope you found this webcast presentation informative. This concludes our webcast. You may now disconnect. Have a great day.